not too long after I started my lab, we started asking questions about gene duplication and protein interactions, and it took some directions that we did not anticipate, and that made the projects even more interesting. So I try to go through the history of that project with you and share some of the results that we had. So the first part is published, and the last part is being revised right now for resubmission. So maybe the papers will come out sometime. Okay, so what we study in the lab is what we call the life cycle of genes. We've done some work on evolution of new genes, not as much as Christian has done, but we did some of that. But here, what I want to talk about is gene duplication. So all genes eventually undergo duplication. Could be just in one individual in the population, and maybe that duplication won't fix, but sometimes they fix in the population and they can be lost again by pseudogenization. And this phase in which you have two genes can last long enough sometimes that you can, the genes can diverge and they can acquire or lose functions, and eventually they can become so different that you can't even tell that they are homologous sequences. And what we're doing some work trying to understand what happens during this time. A lot of work regarding the evolution of gene expression. But what I want to talk to you about today is what we call compensation, or the fact that some gene duplicates can hide the loss of other copies. And we think that this ability of genes to compensate, which is almost 100% when you are just after duplication, because you have two copies. If you lose one, there's nothing bad happening to the cell. So this ability to compensate somehow gets lost with time as diverge. But in the meantime, there's this compensation happening, and this is what we are trying to understand. So is that something important? Should we care about gene duplication? Well, one reason to support the reason why we should study gene duplication is that we find them everywhere. So whatever genome you look at, there's between roughly 20 and 50% of genes that will have a duplicate of really young age or older age somewhere in the genome. So this is true for small genome, like bacterial genome, where you can get up to like almost 50% of genes that have duplicates, and even in across eukaryotes, it doesn't matter if they are unicellular, like Saccharomyces cerevisiae, the bunny that we use, or plants. Lots of genes have gene duplicates, and so this has obviously super important consequences. One of them is that these genes, once mutated, they can acquire different functions. The example I like to give that is quite striking when we teach basic cell biology, I like to tell students about that, is the case of olfactory receptors in mammals. These receptors are GPCR receptors. They're in the cell membrane. They allow us to detect molecules in the environment, and the number of genes within that gene family of olfactory receptors can reach, in some species like elephants, almost 2,000 copies, presumably many of them being able to bind different molecules, which allow the animal to detect, smell different things in their environment. This number changes a lot from one species to the other, and there's some nice studies showing that this is linked to the environment. When mammals went to the sea, for instance, they lost many of those because they're useless in the water, some of them. But for some species, this ability to smell different molecules actually is ecologically super important. What's interesting about this figure also is that you can see in red the number of genes in the family, and in blue the number of pseudogenes that you can find in the gene. We can argue that some of them are still functional genes, but most of them are, most likely true pseudogenes. And so you can see here the life cycle that I mentioned in my first slide, where you have genes that give rise to, duplicates, and eventually many of them, they become pseudogenes. So there's traces of that happening in genome. So you can trace, you can connect gene duplications with many, many things. I mentioned olfactory receptors. You can think about rhodopsins that allow different species to detect different wavelength in their environment. So gene duplication is super important. But what I want to do and what we study in my lab is what happens when gene duplicates, what happens to cellular network. So if we go within cells, uh, can we use the tools of synthetic biology and systems biology to know what actually happened molecularly? And one of the things that we are interested in, like I talked about it on my first slide, is the fact that there's some redundancy between duplicates, which allows them to compensate for each other's loss. So this confers some robustness to cellular networks, and we know a lot about robustness conferred by redundancy. We use that in human-made systems all the time. Like we have two co-pilots in the plane, so if one can't do his work, then the other can compensate for the loss of the first one. So that's what I mean by redundant. And yeast was a great, I mean, people have known about genetic redundancy for a long time. So whenever people in the past have tried to inactivate genes, sometimes they knew the gene they were inactivating was super important, but they realized that there was no phenotype that they could see when they did a gene deletion. And very often this is due to the fact that there's another copy of the gene. So even if the other copy is not 100% identical, it can still buffer the loss of the gene deletion. So this has been known for a long time, but the first systematic demonstration of the role of gene duplicates in conferring resistance to mutations was this study in 2003, where they deleted all the genes one by one in Saccharomyces, in the yeast Saccharomyces cerevisiae, and there's about 6,000 of them. And then they measure whether the effect on fitness was a weak effect or growth rate in the lab, a moderate effect, a strong effect, or lethal. Lethal means you delete the gene and cells cannot grow anymore, even in rich conditions. And then they classify these genes as being singletons, so having no duplicates or having duplicates. And what you can see is that for deletions that have weak effect on growth, you will see a higher proportion of genes in that category that have duplicates. And when gene deletions are lethal, they lead to a lethal phenotype. There's an underrepresentation of genes that have duplicates. So this clearly shows an association between gene duplication and robustness to mutations. This has been extended also to humans, and people have shown that if you measure the probability that on the y-axis here that a given gene is associated with a disease in human, and you plot that as a function of sequence identity of the closest homologue in the genome or the closest paralog in the genome, you can see that this probability decreases. So if a gene has another gene, an homologous gene, a paralog somewhere in the genome that's quite similar to itself, it's very much less likely to be involved in a disease than if there is the closest homologue is quite different. So that means that close homologue has had time to diverge in function. So what we wanted to do a few years back was to try to understand how does it work. So what happens if you have two duplicates that are exactly the same or that have diverged for like a few million years? What happens when you delete one copy? What happens with the other copy? How can you buffer the loss of the first one? Is it something passive happening, or there's something that's changing in the cell to make that happen? And we wrote a review where we tried to classify the different mechanisms that people have identified that could lead to this robustness. And people have noted sometimes that genes change in activity. So if you delete a paralog, its other copies or its paralogs sometimes have higher expression level, or they have, the enzyme could be more active, for instance, if it's an enzyme. What we want to study at the time was binding. So in the protein interaction network, if you delete a paralog, what happens to the binding pattern of the other paralog? And the idea was the following. So if you have a small network here with that red protein that has some interaction partners, right after duplication, these two proteins, they have exactly the same interaction partners. And with time, they will diverge. So one could keep one interaction partner and the other could keep the other interaction partners. They could sub-functionalize, for instance, in terms of protein interactions. And what we asked was, if we go in the cell, when we do a gene deletion, basically what we do is that we remove the blue node from the network. And we wanted to know, do we see buffering at the level of protein interactions, or do we see, or compensation? Do we see this red protein now regaining interactions with this partner with which it could interact in the past? So this would mean that there's active compensation, and this compensation is actually happening at the protein interaction level. So we developed an experimental system with which we could do that. So when I was a postdoc, we studied the yeast protein interaction network using a method that I will tell you about in a few seconds. We identified a few thousand protein-protein interactions among yeast proteins. So everything was done in yeast, and everything I'm going to tell you about this was done in yeast. So we have this collection of yeast strains, which is, about 8,000 strains, for which we have constructs that allow us to measure pairwise interactions between proteins using this assay here on the right. So let's say the two spheres here are the two proteins that you're interested in. You want to know if they physically interact to perform their function in the cell. And what we do to know that is that we fuse to these genes fragments of a reporter enzyme, these two fragments here. And if the two proteins interact with each other, the fragments of the reporter enzyme are reconstituted, and they make the enzyme working. And with the assays that we use, this enzyme being reconstituted allows yeast to grow in a specific growth media. So it's basically coupling protein-protein interactions with a survival assay. And now we have yeast strains so that we can do these test for these pairwise interactions for any pairs of yeast proteins that we want to study. And what's particular about this assay is that because we reconstitute an enzyme that's connected with growth, it's kind of quantitative assay. So if there's more protein complex being formed, yeast will grow faster. And if there's less protein complex being formed, yeast will grow slower. So we can actually measure all protein interactions changes between genetic backgrounds or between mutants of the protein. And at the time, we were doing all of these assays using a robotic platform, using what we call arrays, colony arrays. So what you see here is a plate, just an example of these experiments, where this agar plate is like a petri dish, but it's rectangular instead of being round, and it's about the size of an iPhone, if you want. And all these colonies here, they are specific crosses between two yeast strains that report on one particular protein-protein interaction. So when you see, this is not a random plate, there's some repetitions on the plate. That's why you see these patterns, like these 4 colonies that look the same when they're neighbor of each other. But you can see that these large colonies, they basically tell us that these two strains of yeast that you put at that position, they contain proteins that interact with each other. And if the colony is big, they interact a lot. And if the colony is really small, they don't interact, or they interact weakly. So we can do that systematically. And then we combine yeast genetics with this assay that I just told you about to be able to combine gene deletions with protein-protein interactions. So we could take any motif in the yeast protein interaction network, like this one, where you have, let's say, the red and blue protein again. They are duplicates of each other. They have the same interaction partner. Now we can go in and delete the blue gene and ask what happens to this interaction between the red and the green protein. Does it increase? Does it decrease? What happens? And then we set up a systematic way of doing that using the robot, so we could do that like almost automatically, just by taking collections of yeast strains and combining them with each other. So we did that, and we asked the question that I posed a few slides before, like if we do that with paralogous proteins, what happens? So we set out to do the experiment with around like bit more than 100 pairs of genes. The experiment worked and produced high quality data for 56 pairs of duplicated genes that are single duplicates. So they have only another, like they're basically gene families of just 2 members. And we chose these pairs so that they can represent a diversity of expression and sequence similarities and origins, if they are from whole genome duplication or small-scale duplication. And then we ask, do we see compensation at the level of protein-protein interactions? And if we see that, is there any interesting things happening that can explain why some pairs can do compensation and some others cannot? Okay, so before I show you the data, I'm going to show you the actual types of results that we got, and then I'll show you the data. It's going to be easier to understand. So just to remind you what we do here, we take a protein interaction network. We have two paralogs, red and blue, and they could have like mostly distinct interaction partners or shared interaction partners. We have different patterns in the data. And what we were looking at, and is something we saw, is that if we delete the blue protein, we do see increase in protein interactions, like represented by the thicker lines here that are connecting these proteins. So we do see increase in protein-protein interactions. So we do see compensation happening. I'll show you the numbers in a few minutes. But we also saw something that was surprising. And it turned out to be more interesting. So I'm going to focus most of my talk on the thing that wasn't expected, is that we also saw losses of interaction. So basically, if you delete the paralog, then this copy that's left is also losing, in this case, most, if not all of its interactions with other proteins. So instead of saying compensation, now we're saying dependency. Instead of having two genes that can mask each other's loss, we have two genes that are duplicates and then depend on each other. It's kind of opposite of robustness. Now you have two genes and they cannot replace each other. Okay, what does the data look like? So as I mentioned before, we can measure protein-protein interactions in a quantitative manner. So I'm showing here all the protein interactions that we measured. We measured them in the wild type background that's shown on the x-axis here, and on the mutant background, where the mutant background is the gene paralog that's deleted. And then you can see in general, there's a very strong correlation between the two measurements. In general, there are many, many things that don't change, which is reassuring. So if everything was changing, we would be worried about our assays. And then we see compensation. So those are all these interactions up here that are stronger in the deletion background than they are in the wild type background. And then we have these dependency cases where interactions for a paralog are weaker when we delete its other copy. And those are cases of dependency. So one thing we were wondering about at the beginning was, do we see both compensation and dependencies within a given pair of paralogs? Or do we see like different kinds of pairs of paralogs showing one type or the other? And what we found was the latter. In general, pairs of paralogs, they are compensating or they are dependent on each other. So this is what I'm showing here on this plot. So I have on the x-axis the perturbation score. That's basically deviation from the diagonal from the previous plot that I showed you. It's how much change there is between the wild type and the deletion strain. And then we rank them in terms of average impact that deletion has. So these are all the 56 pairs of genes that we had in our experiments. And then the red dots represent compensation that we detected that were significant, and blue is dependent. And you can see in general that we have three types of pairs of paralogs. We have those that generally show compensation. We have those at the bottom that show dependency. There's some in the middle where we don't actually see anything. There's no compensation, no dependency. So it seems that it's not like every pair of paralog shows some behavior, one behavior or the other. They tend to show one case or one type of response or the other. So this is what this was super interesting, because it means it's not something stochastic happening. There's something systematic happening for some of these paralogs. So I'm going to show you a few slides about compensation, because that's the idea that I implanted in your brain at the beginning. And it turns out it's not super interesting what's happening, but I just like show you what we think is happening. And then I go back to dependency, and I will talk about this for the rest of my talk. So the first thing that people have noticed in the literature, and this is kind of rare, is that but it happens sometimes, is that when you delete a paralog, sometimes the other paralog is more expressed. There's some feedback loop that makes the other paralog more expressed. So if this was happening in yeast, we would also see an increase in protein interactions that would be caused by change in transcription level. We didn't see that. So we looked at data from the literature where people have studied gene expression for all genes in all of the deletion backgrounds. And you can see the results here. So you can see on the x-axis for the genes that we studied and our experiments that were present in the data from the literature. You can see the abundance or expression level on the x-axis in the wild type cells and in the deletion background. You can see the compensating pairs are marked in red. You can see basically there's almost nothing happening. There's nothing happening at the expression level. We did some experiments where we fused the paralogs with GFP so we can measure change in expression if there was any in the deletion background. And we did that for a couple of pairs. You can see here again, like expression in the wild type background or protein abundance wild type background on the x-axis and the deletion background on the y-axis. In general, there's no change. The only cases that are significant are the cases in green, and the effect are so small that without it can explain any changes in protein interactions. So what we think is happening with compensation is the following. So what we think is happening is that in wild type cells, we usually see quantitative changes or preferences in binding. We have some paralogs that bind stronger with some partners than others. And when we do a deletion, these paralogs, they're competing for the same binding partner. So when we do a deletion, we just shift the binding equilibrium towards the other copy. And this is what we call compensation. There's actually not much happening, just like we're just shifting the binding equilibrium. And one of the ways by which we showed that is that we reason that if this is true, if we overexpress the red protein, then we should see a decrease in interaction of this protein, for instance, with the blue protein. And this is what we saw. We did that for a couple of interaction partners, and we saw that. So it's basically a matter of binding equilibrium that we're shifting when we do deletions. Okay, now let's go back to dependency. So just a reminder, when we saw dependency were cases where if you delete the paralog, like the blue protein, which is a duplicate of the red one, we see cases where this red protein also loses its interaction partner. And then we went back to some experiments that people have done in the past and some of ours, and we found out that sometimes they interact with other proteins, and these interactions actually stabilize them. So if you remove an interaction partner, sometimes the protein that remains is unstable and degraded. And we thought maybe this is what's happening with the paralogs. Maybe the two paralogs, they physically associate with each other. When we remove one, the one that is left cannot compensate because it's destabilized. So that's how the dependency would take place. So this is what we looked for in the data, and this is what we found. So if we classify dependent pairs as forming what we call heterodimers, they're basically a dimer, so two proteins that interact with each other, that are the two duplicates. So we call them heterodimers of paralogs or paralogous heterodimers. We can classify them as heterodimers or not forming heterodimers among the dependent pairs and independent pairs, the ones that don't show dependency. You can see I mean, this was partial data at the time, but we have more data on that now. It's still the same. We can see that dependent pairs, they tend to be formed by heterodimer paralogs, and independent pairs, they don't form these heterodimer paralogs. Now, okay, so this is probably the mechanism by which this happens. And why is that interesting in terms of evolution? I'll tell you that in a few minutes. So before I go to the evolution part, I just want to show you so that we confirm that these physical interactions were probably stabilizing the proteins by measuring protein abundance again. So in this case, what we do again is tag the proteins with GFP and measure their abundance in the wild type background on the x-axis and in the deletion background on the y-axis. And you can see all the red dots, they are for dependent pairs, and the stars, they are cases where we measure and is a significant decrease in abundance in the deletion background. So all cases. And so this was done by flow cytometry. We can do the same by standard Western blot that people have used for a long time to measure protein, like changes in protein abundance, just to validate our results. And we can do that here. You can see in this case, this is a case where deleting the paralog doesn't change abundance. So you can see the band of that protein is the same in wild type and the deletion background. But in some cases, when we delete the paralog, the protein we're focusing on is almost inexistent in the cell. And we see other cases like that. So clearly, the fact that these proteins interact with each other does something and makes them unstable or makes them being degraded when we delete the paralog. Now, okay, why this is interesting from an evolutionary standpoint? So this is interesting because the way that you get two paralogous proteins to interact with each other is through the duplication of one particular type of gene. And these genes are genes that code for homomeric proteins. So if you have a gene that codes for a protein that interacts with itself, like this one here, after duplication, what you get is that you get two complexes. You get that, this protein interacting with itself. Let's say the other paralog coding for a protein that interact with itself. And you also get the heterodimer. So when you duplicate this type of protein, you get three complexes instead, and you get one for free. You get this new complex here that didn't exist before, and that's made by the interaction of the product of the two duplicated genes. And what our data suggests is that when this happens, these two proteins, they can interact in a way such that the two paralogs, they cannot compensate for each other's loss because their coevolution makes them codependent. So following the duplication of this type of gene, you can have this kind of pattern happening, like coevolution that prevents buffering by gene duplicates. So we tried to verify whether this was actually the way it happens with the paralogs that we find in Saccharomyces cerevisiae. And one way to do that is to try to infer the ancestral state of these proteins. It's hard to know if a protein in Saccharomyces cerevisiae was forming an homodimer in the ancestral fungus. But one thing we can do is that we can use data from a not too far related fungal species, Schizosaccharomyces pombe in this case, and see if we look at, for instance, all the paralogs that come from the yeast whole genome duplication that are called homolog, and we can see when they form heterodimers. Do they have orthologs in pombe that were homodimeric proteins? If they were homodimeric proteins, we can infer that they were probably in the last common ancestor. They would probably also be homodimeric proteins. So what happened after duplication was this pattern that I showed you before, that the two paralogs would now form heterodimers. And this is exactly what we saw. So these heterodimers of paralogs, they tend to originate from ancestral homodimers. So here, what I'm showing on this plot is the percentage of orthologs in pombe that form homodimers for the different categories of genes of whole genome duplicates we can find in yeast. They are like the ones that are not duplicates, the ones that that are duplicates from the whole genome duplication, homologs, and the ones that are homologs but that form heterodimers. And you can see that there's a strong enrichment in this category for homologs or orthologs in pombe that form homodimers. So what we think is happening during evolution, there's something interesting happening, is that you have ancestrally, before duplication, you have these proteins for which the functional unit is an homomer, like a protein interacting with itself in two copies or multiple copies. We don't know most of the time, but it does happen like that. And then what happens after duplication? These two paralogs interact with each other, and in some way they co-evolve such that, and I'm going to tell you all this happens in the second part of my talk, they interact in such a way that if you go in yeast cells, you delete the paralog, now the other paralog is no longer functional. So these two paralogs, now they became codependent, or they became the functional unit. So you had before one gene, one protein that was doing something. And now you basically have two genes, two proteins, but one complex that's presumably doing something very similar, or maybe even exactly the same thing. So replace something with one gene one function to 2 genes, 1 function. So instead of having duplicate now that can buffer each other, you have duplicates that became dependent on each other. So this is what we're trying to study now experimentally. So we're trying to see this transition. How can this happen, and what are the consequences? And the reasons why we think this is super interesting is that you have a case where you have a duplication that doesn't necessarily lead to more robustness, because now these paralogs, they're not redundant. And the other reason why this is interesting is that you can have more genes, but basically potentially have no gain in function. You just replace a simple machine that works with 2 copies of itself, and now it copies of 2 different copies of the proteins that are coded for from different genes to do the same work. So in the next part of my talk, I would like to convince you that this is probably something important, and I want to show you how this can potentially happen. So one of the reasons why we think this is something that could be interesting is that these homomers, like proteins that interact with themselves, they are a large fraction of all proteins. So if you look in protein structures that have been studied in vitro, like in the protein data bank, so that dates from a few years back, which is actually the numbers don't change much with new data or with AlphaFold predictions. You can see in unicellular eukaryotes, which basically means yeast, and vertebrates, which most of the time means humans in these types of analysis. You can see that homomers, like proteins that interact with themselves, they make up for about almost 30% to more than 40% of all proteins. So it means that there's like 30% of genes that, when duplicated, they create these heterodimers of paralogs. So they create the possibility of this coevolution between paralogs. And a few years back, we measured this in vivo in yeast, and we look at different categories of genes and the gene duplicates. It doesn't matter really. What matters here is that it's about 30%. So what we measure in vivo using the technique that I showed you before is basically what's been shown in vitro. So this is something super important that could take place, every 3 times there's a gene that duplicates. There's one of those 3 times is a gene that codes for a homodimer that leads to this new complex between gene duplicates. Okay, so when we go out of yeast and we look at larger scale comparisons that people have done, like this paper here by Pereira Leal et al. In 2007, they already had noticed some trends in that direction using long-term comparisons of protein structures. So very often, it depends on how it's very subjective. But let's say it happens quite often that you have, for instance, in E. coli, an enzyme that works as a dimer, or homodimer. So the 2 proteins are the product of the same gene. And when you compare another species like yeast, these enzymes now, they work as heterodimers. So the 2 proteins that assemble, they come from paralogous genes. So it means that for many, on the long term, this actually what we saw and we proposed in terms of model to explain the dependency between paralogs is actually happening. So you have this duplication happening, and now for a while you have these 3 complexes, and eventually there's this complex that remains, that is doing the job that, as far as we know, in most cases that people have studied, this enzyme in yeast doesn't work any better than it did in E. coli or vice versa. So there's not necessarily, there's some reasons to believe that this could be advantageous, and I'd be happy to talk about that at the end if you want, but let's assume, given the data we have, that there's no reason why this would be advantageous. So in that second part of the talk, so for the last 10, 15 minutes, I want to tell you how we try to reproduce that in the lab and see how it can actually happen. Because we can, on the long term, it's really difficult to understand what happened between, the common ancestor of E. coli and yeast, and on the branch leading to yeast or the branch leading to E. coli. It's really hard to reconstruct events and know what happened. So what we thought we could do is try to test that in the lab and see if we can evolve that in the lab, so we know exactly what happened. A bit like you guys do in Christian's lab, where you perform experimental evolution, you have time points, you know what happens when, and you can better control things. So the model we set out to test was the following that could explain what we see in the data. So we have an ancestral homodimeric protein, and after duplication, we have two gene copies, A and B. And now, because you have two gene copies, a loss of function in the first paralog that makes paralog A non-functional, produce a complex that's non-functional. This loss of function would be neutral because there's this additional copy that can perform the function. But now what happens is that you have a different genetic background. And in this particular genetic background where A is not inactivated by loss of function mutations, now there might be some loss of function mutations in B also that, when combined with the loss of function in A, would create a functional complex. So this would be like positive genetic interactions. You have two loss of functions in A and B, and when they're combined, they reconstitute, they kind of compensate for each other's loss of function, and they reconstitute a complex that's functional. So if this is possible, then you have the first loss here that is neutral, and this mutations here would be neutral in the background of the first one, because it creates a complex that's now functional. So this is one way by which we call these mutations compensatory mutations. So they compensate for each other's loss. So you could go from a situation where you have an ancestral protein that's an homodimer, and you place it with an heterodimer by combining the compensatory deleterious mutation. So what we set out to do was, let's study that. Let's see how many mutations we need to get there, how active this complex is, and what would be the molecular mechanism. And so we went to the lab and we did that. And we found a model that's called, with which we could test this question, that's called a cytosine deaminase. So this protein is called FCY1 in yeast, but it's a protein that's shared with other fungi. And it codes for an homodimer, so you can see that the two copies of the protein here. And what's nice about this protein is that we can do positive selection on this protein, because in specific growth media, it can convert cytosine to uracil, and we can make this reaction essential. So we can perform positive selection on mutants of this enzyme. And we can also perform negative selection on the function of this enzyme using this drug here that's called 5-fluorocytosine. And this drug is inactive, and it's made active by the activity of this enzyme. It becomes, through deamination by this enzyme, it becomes 5-FU, and 5-FU is toxic for yeast cells. So we can also do negative selection on mutations. So we can provide higher fitness to loss of function mutations. You'll see in a few minutes why this is the trick that we needed. So to do our experiments to identify mutations that could compensatory mutations, what we did was an experiment we called deep mutational scanning. Many people are doing that in different fields, and genome editing. Basically, we take the yeast gene, we create a library of all possible single mutant in this gene, and then we take the library and we introduce it in the yeast genome using CRISPR-Cas9 editing. And now we have a library of yeast mutants, each differing by a single amino acid substitution in this enzyme, and we can combine them and then perform selection experiments, a bit like you guys do on Drosophila, and then we can follow the allele frequencies through time, and alleles that are deleterious would decrease in frequency, and those that are beneficial would increase in frequency. And all we can do is that we can put this library in a condition where we select for function with positive selection, or where we select for loss of function and apply basically negative selection on the function of the enzyme. I'm going to show you just the kind of results we get from this. This is just to show you we cover mutations, and there's a lot of loss of function mutations in that enzyme. And then I'll show you how we can use that to study gene duplication. So what I'm showing here is the kind of results we get when we do these experiments. And what I'm showing here is just resistance to 5-FC. So that's basically loss of function. When it's yellow, it means it resists that toxic molecule, it means that the enzyme is dead, basically. And so you have all the positions of the enzyme on the x-axis, and on the y-axis, we have all the amino acids that we introduce at each of these positions. And the red dots represent the wild type amino acids at these positions. You can see that if you follow the red dots, you see that all these like wild type sequences, they are sensitive to the drug. And all the ones that are resistant are shown in yellow here. And you can see at the bottom represents top codons we had in our library. They are all resistant to 5FC, except a few at the beginning. I can explain why if you're interested in a few minutes. And we can map these mutations on the structure. Again, I'm just showing resistance here in yellow. You can see this is the homodimer again. You can see inside there are many yellow regions inside the protein and at the interface. And those deleterious mutations, the vast majority of them, they are mutations that destabilize the enzyme. So they make it non-functional. So the cells resist 5FC. So now we know there's a lot of loss of function mutations in this enzyme, single mutations that leads to loss of function. So what we can do is that we can take our libraries, introduce them in the two yeast mating types, A and alpha, which are basically two sexes in yeast. And then after that we can do crosses. So we take our two mutant libraries, we apply negative selection. So these libraries of mutants will contain all the yellow mutants I showed you in the previous slide. So we have enzymes that are not working here, enzymes that are not working here, and then they would represent these loss of function mutations. And after that, we can mate them, and we can apply positive selection. And then any combination of two loss of function mutations that compensate for each other, that create a functional complex, would now increase in frequency in that condition. And we did that, and then we plated all these positively selected clones on plates to select the ones that are working, isolated them one by one, and by high throughput multiplexing, we sequenced them. So we have pairs of mutants for the two paralogous genes. So just to show you a little bit of numbers, so we found compensatory mutations at all of these positions of the enzyme that are marked, in the first paralog, we call A, and in the second paralog, we call B. And the numbers here are the number of times at each of these pairs of positions for which we found compensatory mutations. And we see these hotspots here, where at the catalytic site, there are many, many mutants that are capable of compensating for mutations at other sites in the other gene copy. And we have many mutations also at the interface of the dimer. So these are two patterns where we see a lot of compensatory mutations. So we can put them on the structure. Here in yellow on one side and red on the other side, you can see compensatory mutations. So the active site of the enzyme is here. And you can see there's some mutations around the active site and many that are near the interface. And we can connect them between the two. Let's say this is our heterodimer, so the two paralogs, and we see this pattern of compensation. We can see these mutations at the active site in this region. They are compensated by mutations at the interface, for instance, very often. And we also have pairs of compensatory mutations at the interface, like one-to-one mapping. So I'm gonna just show you for the last few minutes data on the functional consequence of these mutations. I'm gonna focus on one example, this this pair of mutant here, which is a mutation at the catalytic site and a mutation at the interface here. Yeah, before I go there, I want to, one of the things we wanted to make sure is that the clones, the pairs of mutants we found were actually at a fitness, a growth rate that was similar to the wild type enzyme. So after duplication, once we have these compensatory mutations, we wanted to know what's the fitness of these mutants. So we recreated many pairs of mutants and measured their fitness, and we made crosses among mutants that were not found to be with each other. And then what you can see is that distribution of fitness values that we saw compared to the wild type. You see that we have many pairs of compensatory mutants that give fitness that are in the range of the wild type mutant. And most of the pairs we didn't see, are not compensatory. And we saw some by chance that were also compensatory in the experiment. So one thing that we wanted to make sure was that we're measuring all of these things in yeast. Maybe there's something happening in yeast that can make some of these mutant when we combine them functional, and it has nothing to do with the assembly of the heterodimer. So now what we have, I'm not going to show you the data. We have done experiments to show that the heterodimers are forming in yeast. We also have crystal structure of the heterodimers that are in the preprint, some others that we're working on in the revisions for the paper. But one thing we wanted to do was to actually purify the heterodimers and measure its activity in vitro, make sure that the enzyme is actually working. So this brought us to more protein biochemistry, where we basically clone the alleles that we had in an expression system in E. coli, and we purified the complexes, and we could measure using plate readers the conversion of 5-FC to 5-FU, and that gives us some measure of enzyme activity. So we can do that with single mutants, or we can combine mutants together, see if when you combine them, you get better activity. So what you're seeing on this plot is just like a time course measurement of this conversion here. And on the y-axis is the amount of conversion that we see. And for mutants that we uncovered, including the ones I told you about that I marked on the structure, which is this E64V, the catalytic dead mutant, and this R73D, which is a mutant at the interface. You can see if we express all of these genes alone, these mutants alone, we don't see any activity at all. We cannot detect any activity. If we combine them, then we can start seeing activity. So the y-axis is, again, is relative to the wild type. So we don't see exactly the same activity as the wild type enzyme. There's some reasons for that I can tell you in a few seconds. But you can see if we combine, for instance, E64V, which is the catalytic mutant, with this interface mutant, R73G, now it gains its activity. So the single copies were dead. When we combine them, suddenly we get an enzyme that's working. So it's truly compensatory mutations. So what we see for this pair of mutant, we have other patterns now that we can extract from structure, but I focus on this one because it's a bit simpler. We have these mutants. So alone, they cannot work. So they kill the enzyme. When we combine them, we reconstitute a functional enzyme. So what's happening here is that we know what's happening. So this residue E64 is the catalytic site at the catalytic site of the enzyme. So it kills the activity of the enzyme. But what happens when we measure its stability? It's a protein that's now more stable. So it's very often there's a trade-off between stability and activity in enzymes. And if you remove active sites, often you get a more stable enzyme. So this protein now is dead. It's not active, but it's more stable. And this one, what happens is that you have this substitution at the interface that makes it unstable. So it would be active, but when it's expressed alone, it's unstable and the protein is not functional. But if you express it with E64, it creates this complex where this one provides stability, and this one provides activity. So we have these two compensatory mutations. One brings in stability, the other brings in activity, and now you go back to the function you had before through compensatory mutations. And that looks a little bit like what I showed you in the first part of the talk, although the first part of the talk, I didn't focus on enzymes, but here we have two gene products that are from gene duplications, independent copies, but they cannot buffer each other's loss. They actually need each other to produce a functional protein complex. Okay, so this is where we are with this project. There's more details on the structures in the preprint. But let's see if we can summarize everything we've saw today, and maybe that's gonna hopefully influence the way you think about paralogs in the future. So paralogous genes, they can buffer each other. This has been established for a long time. But what we show that was novel here, and the extent of it, is that they can also become codependent. And so that was the first part of the talk. In the second part, what I showed you is that we can actually produce this dependency in the lab. And it doesn't seem to be super difficult to do. You just need one mutation per gene, and that seems to be sufficient to turn homodimers, like homodimers into an obligate heterodimers of parallax. And the imaging pattern we saw, but there's other interesting patterns that we've seen also with the crystal structure, is that there's one subunit that performs the enzymatic function, and there's another that's stabilizing. So it's like if the two copies, they sub-functionalize, but they sub-functionalize at the molecular level, and together they form a complex that's functional. So this is the framework that we're working on now when we study gene duplications. That's what we want to study. Now we have the pattern that we have in mind when we study gene duplication. We go from one copy, two copies. The two proteins, they can accumulate their own mutations. They are in their own like fitness landscapes, if you will. They can evolve in that space. When we have a duplication of an homomeric protein, things are different, because then now you can have mutations in one paralog that's gonna affect the way mutations impact the second paralog, because these two proteins interact with each other. So we have a couple of projects now trying to understand how this physical interaction actually impacts future evolution of duplicates. So if this is something you're interested in, we have like a paper where we simulate this type of evolution that came out. It's more about everything in there is computational, but that's based on actual protein structure, from which we try to see whether mutations would favor this type of evolution on the long term. Okay, so this is the end. Just want to mention who did the work. So the first part of the work was initiated by Guillaume, one of the first PhD students in the lab. Is now a faculty, a dependent researcher in Basel. Philip, the second part was done by Philippe Després, who's just finished his PhD last year, is now a postdoc. And a lot of the work was also done by research associate Isabelle and Alex, who've been working with me since I started my lab. And all the structural part at the end, to do that, we teamed up with a structural biologist from our institute, who's been working on trying to understand molecularly how these compensatory mutations can take place, and how at the structural level we can explain what's happening.